In controlled environment agriculture, it's common to see pink or purple glows from red and blue LEDs. These are sufficient for plant growth with high photosynthetic efficacy, which is described as micromoles of photon output per joule of energy spent. For indoor vertical farms that rely on electric lighting, high fixture efficacy is beneficial to operating cost management and crop profitability. The ratio of red to blue light influences plant extension growth, yield, and secondary metabolism. For most plants like lettuce, increasing the fraction of blue light decreases leaf area and biomass. On the other hand, more blue light produces more nutritionally relevant secondary compounds such as anthocyanins, which are the red pigments in red leaf lettuce. There's one caveat though about red and blue LEDs. They don't reveal the true colors of plants, so it's difficult to inspect plants for pests, diseases, and nutrient disorders. If we add some green light, we get white light. That makes it easier to see the plants. Research has shown the usefulness of green light for plants, even though green LEDs are still very inefficient. In recent years, there have been growing interests in using broad spectrum lights to grow indoor specialty crops such as leafy greens. Broad spectrum lights are white LEDs or a combination of white, red, and blue LEDs. White LEDs are essentially blue LEDs based on a semiconductor material called indium gallium nitride. The blue LEDs are covered with a phosphor coating that converts high-energy blue photons to lower-energy green, red, and far-red photons, hence the term broad spectrum. The combination of blue, green, and red light appears white to our eyes and reveals the true colors of plants. The phosphor conversion of photons decreases fixture efficacy. In general, there are three types of LED grow lights in the market. First, we have red and blue LEDs. The spectrum shows two distinct peaks near 450 nanometers for blue and 660 nanometers for red. Lower blue light around 10% is generally marketed for faster vegetative growth, whereas higher blue light around 20% or more can increase secondary metabolites. Secondly, we have white LEDs. The spectrum has a peak around 450 nanometers for the base blue LEDs, and another peak in the green or red region for the phosphor-converted photons. Depending on the phosphor material, more or less blue light is converted to photons at higher wavelength. Warm white LEDs have less blue light and are less efficient than cool white LEDs. The spectrum in the middle is for specialized mint white LEDs that emit a high percentage of green light. Thirdly, we have white LEDs mixed with red and blue LEDs. The spectrum shows the white LED spectrum superimposed with additional red and blue spectra. This approach strikes a good balance between color rendering and fixture efficacy. In some cases, far-red LEDs are also included in all three types of LED lights to control flowering or increase plant extension growth. Our research question was, how do plants grow under different white LEDs or white LEDs mixed with different amounts of red and blue light? First, we wanted to compare lettuce growth under warm white LEDs versus mint white LEDs. Mint white LEDs emit more blue and green light, whereas warm white LEDs emit more red and far-red light. All lighting treatments were kept at the same total photon flux density of 180 micromoles per meter squared per second. We postulated that lettuce will grow similarly under the two white spectra. Secondly, we wanted to evaluate the mint white spectrum with supplemental blue and red LEDs at 2 blue to red ratios. We partially substituted mint white LEDs with 80 micromoles of blue and red LEDs at a ratio of 1 to 7 or 5 to 3. From left to right, blue light increases. We postulated that increasing the amount of blue light would decrease lettuce growth but enhance red leaf coloration. Thirdly, we wanted to compare lettuce growth under mint white blue-red LEDs to blue-green-red LEDs with similar photon flux densities in each 100 nanometer waveband. For example, the combination of 100 micromoles of mint white, 10 micromoles of blue, and 70 micromoles of red has around 20 micromoles of blue, 60 micromoles of green, and 100 micromoles of red. So we created a combination of the individual RGB LEDs with similar RGB photon flux densities. On the other hand, a combination of 100 micromoles of mint white, 50 micromoles of blue, and 30 micromoles of red has similar RGB light as a combination of individual RGB LEDs at 60 micromoles each. We postulated that as long as the RGB photon flux densities were similar, lettuce would grow similarly under the two LED mixes. We conducted this experiment at the Controlled Environment Lighting Laboratory at Michigan State University. We germinated plants of red leaf lettuce rosé in rock wool and grew them in a vertical deep flow hydroponic system under different LED treatments. The photo period was 20 hours. The total photon flux density from 400 to 800 nanometers was 180 micromoles in all treatments. The average air temperature was 22 degrees Celsius. The average relative humidity was 35%. The nutrient solutions were made of deionized water, a 12,4,16 base fertilizer, and magnesium sulfate. The nutrient solution pH averaged 5.8. The EC averaged 1.8 millisiemens per centimeter at an initial nitrogen concentration of 150 parts per million. CO2 was at the ambient level. Now let's review the 6 lighting treatments. First, there were warm white and mint white. In addition, mint white was partially substituted with 2 ratios of blue and red. Lastly, for each combination of mint white, blue, red, there was an RGB analog with similar RGB photon flux densities. From left to right, the 6 treatments had increasing blue photon flux densities from 12 to 62 micromoles. These were the actual measured values with the spectral radiometer. We performed the experiment twice over time and pulled data from 2 replications for analysis. We collected plant growth data on day 33 in the first replication and on day 30 in the second replication. Here are the data for shoot fresh mass in columns and shoot dry mass in dots. Yield was similar on the warm white and mint white, despite their spectral differences. When mint white was partially substituted with blue and red, yield decreased with increasing blue light. Finally, yield was similar on the mint white, blue, red, and their RGB counterparts with similar RGB photon flux densities. Here, we have shoot fresh and dry mass plotted against blue photon flux densities of all 6 treatments. In general, increasing the blue photon flux density decreased yield. Plants grown under the 2 treatments with the most blue light had 3 fewer leaves than the other treatments with less blue light. This indicates that high blue light slowed down leaf development. For leaf size, we measured the length and width of the 5th most mature leaf. Leaf size was similar under warm white and mint white. Increasing supplemental blue light to mint white decreased leaf size. Leaf size was similar under mint white, blue, red, and their RGB counterparts. Across all 6 treatments, leaf length and width decreased with increasing blue photon flux densities. This confirms the role of high blue light in suppressing leaf expansion. Plant diameter showed similar treatment responses to leaf length. The higher the blue photon flux density in the broad spectrum, the smaller the plant canopy coverage. This can reduce the amount of light captured by the plant for photosynthesis, and thus decrease biomass accumulation. Plants under the 6 treatments showed different degrees of red leaf coloration. From left to right, red pigmentation intensified. This was quantified with A star, which indicates greenness or redness in color analysis. A higher A star value indicates more redness, whereas a lower A star value indicates more greenness. Leaf redness was similar on the warm white and mint white, and was also similar on the mint white, blue red, and their RGB counterparts. The color differences were attributed to the blue photon flux density. Although we didn't measure anthocyanins directly, more blue light likely produced more anthocyanins and thus increased leaf redness. This time-lapse video shows lettuce seedlings grown on the warm white, mint white, and mint white supplemented with 2 ratios of blue and red. 1 second of the playback shows 5 hours of real life. The treatment on the right with the most blue photons produced redder leaves than the other treatments early in the seedling stage. This persisted into the later exponential growing phase. In conclusion, lettuce growth was similar on the warm white and mint white LEDs. For mint white supplemented with blue and red LEDs, increasing the blue photon flux density decreased lettuce yield and leaf expansion, but increased leaf red coloration. Lastly, as long as the RGB photon flux densities were similar, lettuce grows similarly on their mint white, blue red LEDs and their RGB counterparts. We'd like to thank our funding agencies, industry partners, and colleagues who provided generous support to this project. Thank you.